So till now, what we were actually doing was to analyze the existing protein structures that were already available in PDB and try to gain maximum information from whatever structural analysis has been already conducted on say some already studied proteins as such. Okay, what can you extract from an existing structure? What things you can do on whatever structures that are resolved till now, or whatever structural knowledge is available till now. Okay, that was the major focus of discussion till now, and that we did it in terms of modeling the protein structures as such. Okay, now we would move on our discussion towards another fact that sometimes it might entirely happen that you might be interested in say some structure of say some protein whose no one has determined the structure of that protein using X-ray crystallography or any experimental method, but now your guide says, or your PhD mentor or your PG mentor says, that we need to have some idea of the structure of say certain protein. Okay, so in that case it becomes easier for you to have functional insights on say certain proteins whose structure is not resolved or whose structure is not solved by X-ray crystallography. In that case you can predict the structure of proteins. Okay, so prediction is one of the way you can have insights into the structure of the protein, as well as eventually you can have insights on the function of that protein. Okay, so now one would ask why do we bother with predicting the structure of a given protein at all when we can experimentally determine it, right? If experimental methods are indeed available for finding out the structure of a given protein, then why do we go for predicting its structure? Okay, so well, let us first try to discuss why predicting the structure is need of an hour, or why is it important. Okay, so to begin with, I would like to say that there are many bottlenecks in experimental determination of a structure. Okay, which makes experimental structure determination a very long and tedious task. Okay, so it includes multiple processes and it includes multiple steps, like isolation of proteins. Okay, so well, say for example, if your protein is expressed at very lower rate in a cell in a very limited time span, then how would it become possible for you to even isolate that protein from the cell culture? Okay, isolation of proteins sometimes is a difficult job. Okay, first being that not all proteins are after their expression exposed to cytosol. Most of the proteins are membrane protein, and how do you isolate that membrane protein without bringing them out of membrane, right? And if they come out of membrane, eventually their conformation is going to change, right? So isolation of protein in itself is a very difficult task. Okay, and after isolation, not every isolated protein can be purified in a larger amount so that it can be used for crystallization, right? So purification is also one of the very daunting issue if you want to solve the structure of a given protein experimentally. Okay, and finally, not every purified protein would be crystallized, because each and every protein has a specific crystallization condition, right? And it is not universal that okay, this is the crystallization condition in which each and every protein will get crystallized, so that you can collect the diffraction pattern and create that electron density map and create and generate that beautiful model, right? So crystallization condition of each and every protein is different. Okay, and majority of time as well as efforts of any protein solving group goes in these three steps, and most prominently in crystallization step, finding out what are the optimal crystallization conditions for my given protein as such. Okay, so owing to these bottlenecks, the experimental task of determining the structure of a given protein becomes very prolonged and very difficult task. Okay, so these are very daunting bottlenecks that scientific community are still facing. Okay, and moreover, the rate at which the sequences are being deposited in the sequence database is far more higher than the rate at which we are able to generate experimental crystallographic structures. Okay, and due to these differences in deposition of sequence data into the sequence database, and the low number of crystallographic or structural determination, well, it creates a kind of gap between the structure information and sequence information. Okay, so we say that we have enormous sequence information, but we still lack, or we still lag behind in structural information of a given protein or a set of protein as such. Okay, so now if we predict the structure of the proteins, it might help us fill this information gap between sequence and structures that has been generated recently. Okay, so owing to this fact, it is first, it is very easy to predict the structure in comparison with experimental procedure. Okay, and second, it would require less time to do so. Okay, and moreover, there are a lot of studies that are published in very good journals that justify that the predicted structure as well can give you as important insights into designing drugs or understanding the function of a given protein as well as good as your experimental structure does. Okay, so owing to all these factors, it becomes very important for a common biologist to learn these techniques about how protein structures can be predicted, and what we can gain after what we can learn when we determine or predict the structure of a given proteins in silico. Okay, or using computers or using computational techniques, right? So this is how it is important that you should learn how to predict the structure. Okay, so now let us first begin with the overall approach of this protein structure prediction. So do you now get key where exactly we are moving on in our discussion now? Till now we have discussed, till this point we have discussed how protein structure can be visualized and analyzed, right? But now onwards we would talk about how we can how we can determine the structure of a given protein using computational techniques. Okay, that would be the major focus of our interest now. Okay, so this particular figure that you are currently viewing is a typical logic or typical approach that is usually implemented in predicting the structure of a given protein. Okay, बात समझ में आ रही? Now first I will just brief you on what will happen in each and every step, and in our subsequent lecture we would go in more details of each and every step that has been mentioned over here, right? So this is what I have planned for you people in this lecture and upcoming few lectures as such. Okay, so now अपना क्या ऑब्जेक्टिव रहेगा अभी? What will be our objective now? We are supposed to predict the structure of a given protein provided that its structure is not already present, or it is the structure of that protein is not already being experimentally solved. Okay, and how you can do that, right? So first we will go into theoretical details of it. Okay, and since I have been assigned a very few lectures are left with me to discuss these things with you people, it won't be possible for me to uh, deal with the practical aspect. Okay, so anyhow, to begin with, if you want to predict the structure of a protein using this computational technique, people usually first perform sequence based analysis. Okay, so the way to figure out the structure of a protein begins with analyzing its own sequence. Okay, so the very first prerequisite for you to deduce the structure of any given protein that you are interested in is the availability of its sequence. Okay, so it is very important that you know the sequence of a given protein. It is very necessary that someone has already sequenced the given protein. Okay, there should be experimental evidences that okay, this might be the sequence of my given protein. Okay, so
Sir,
yes,
sir, sorry to interrupt, but I think two students are waiting out to be admitted.
is it?
Yes, sir.
Okay, just a minute. Okay, I think they are in now. Okay, here
Yes.
we go again.
Thank you,
So
sir.
it is necessary that you have experimental evidences that this is the sequence of my given protein whose structure I am supposed to predict. Okay, so how do you begin the protein sequence analysis in order to determine its structure? Yes, of course, you can first do this database searching. ओके सबसे पहले आप शुरुआत क्या करेंगे यू वुड डू डेटाबेस सर्चिंग ओके फॉर डेटाबेस सर्चिंग you might use tool like blast. Okay, now what you can do is that you can well say for example now you have the sequence of a given protein. Okay, this line would represent the sequence. Actually it is what it is, well some it might have some set of amino acid residue over here. So for example proline, glycine, alanine, arginine. ठीक है, ठीक है, aspartate, glutamate, so on and so forth. So glutamate is E. Okay, so whenever I represent a line, तो इसका मतलब क्या रहेगा, you are supposed to consider it, you are supposed to consider it what you are, whenever I draw such a line you would consider it as it is a sequence of a given protein. Okay, so by using techniques like a blast, what you can do is that you have such a sequence of your protein whose structure you need to determine. You can search if there are other proteins in your database that have similar sequence. Okay, well say for example if I want to figure out the structure of an enzyme called as homoserine kinase from, let's say for example Leishmania major, right? Okay, now what is our problem, that I want to figure out what is the structure of this enzyme called as homoserine kinase from organism Leishmania major, and in some database I figured out that there is an sequence already given over there. So what do, I download that sequence. Okay, and that sequence would be represented as a line here on. Okay, so now what I do this BLAST. Are you people aware of BLAST?
Yes, sir.
Okay, so what do we do in BLAST? We try to figure out if there are similar sequence already submitted in say some database. Okay, so after doing this BLAST search, you might end up with multiple sequences of the same enzyme. Okay, well, so say for example, this can be homoserine kinase sequence of say Leishmania donovani. You can have homoserine sequence of Trypanosoma brucei. You can have homoserine kinase sequence from yeast. You can have say some from bacteria, Methanococcus jenici, will say for example. In this way, after performing such database searching, you might end up finding out if there exist some set of sequences, or if there exist certain sequences in that database which are highly similar to mine. Okay, now what are these? These are all what? These are all homoserine kinase sequences from different organisms that people have already sequenced and deposited. Okay, is it clear till now? यहाँ तक समझ में आ गया?
Yes, sir.
Okay, so now
Yes.
once you perform this BLAST search, okay, the next thing that you might do is that you might perform this multiple sequence alignment.
Uh, Hello.
Huh? Yes. Anyone want to ask anything?
Sir, Srushti and Keshav are trying to join. They are not. Their request is not admitted.
I mean, I don't understand why don't you people come in time, and everything is being recorded now. Okay, please try to come in time, guys. ठीक है. So where were we? We have identified these multiple sequences that might be, related to the sequence whose structure is needed to be determined in our case. Okay, so what thing next we do is that whatever sequence we have obtained in this way, we would perform multiple sequence alignment using softwares like Clustal. Okay, are you aware of this software, Clustal X or Clustal W? Okay, so you can use this Clustal software to perform what? Multiple sequence alignment. And what would be the objective of multiple sequence alignment? The objective of such multiple sequence alignment would be to figure out, well, say for example, if these are the 5 sequence now with us. The first one is from Leishmania major, then Leishmania donovani, then yeast, then Methanococcus jenici, okay, and we'll say Trypanosoma brucei, right? So these are all homoserine kinase sequences you have ended up identifying from this BLAST. Okay, so what we'll do next in this multiple sequence alignment is that our objective would be to figure out how much portion of this protein has remained conserved throughout the evolution. Okay, so well, say for example, if this is the start of your sequence and say first 50 amino acid residue are highly conserved. When I say highly conserved, that means well, say for example, this would be the sequence in all of the homoserine kinase in all of the organism that we are considering. Okay, if such is the case, we say that this particular sequence is highly conserved throughout all of the sequences. So in this way you can figure out in your unknown sequence, कौन सा unknown sequence, Leishmania major from homoserine kinase homoserine kinase from Leishmania major, that okay, from 1 to 50, this particular sequence is highly conserved throughout the evolution. And we'll say, for example, next 5 or 10 amino acid residue, okay, there might be certain variation in this region. Okay, while the other next, we'll say, for example, if it is 300, then to 90 ke aspas, we'll say, for example, this sequence is again highly conserved. Okay, and this sequence is variable. So now you end up with some information like whatever structure of my protein would be, structure of what? Homoserine kinase from Leishmania major. Okay, so in this structure, this much sequence is highly conserved, while this much sequence might have certain variation, while this much sequence is again highly conserved throughout the different homoserine kinases that are known so far. Okay, so now you know our objective is to find out the structure of this Leishmania major. And now by doing this multiple sequence alignment, what have we figured out? Which is the region from my protein which is highly conserved, and which is the region from my protein which is not conserved, or which has got sequence variation in it. So this much sequence, or this much portion of my protein will have variation. It do not match with other proteins as such. Okay, so we are going to use this information later. Okay, so once you know which region of my protein is highly conserved and which region might have variation, we would use this information later. Okay, so now what we will do is that we would ask a question in PDB. Okay, we would ask a question: if the homologue of the given protein, if the homologue of homoserine kinase from Leishmania major is present in PDB or not. That would be our question. Okay, homologue यानी क्या? Some protein who are evolutionarily related to the given protein.
Each other.
Okay, and we are specifically asking that if the homologue is present in PDB, right? तो PDB में है मतलब क्या? उसका structure already available है, right? So if the answer is yes over here, so what would it mean? It would mean that one of the homologues that we have identified, its structure is already present in PDB. Okay, and if that is the case, then you can use the technique called as homology modeling. Okay, homology modeling को कब जाना है? When you know that the structure of one of its homologue has been already experimentally determined. Okay, now you have the sequence. Now you have the sequence of your protein, and the structure of its one of the homologue has been already determined using some experimental technique. If you have these two situation, if you have the answer to these two question as yes, then you can move on and use the technique like homology modeling. Okay, and using the technique like homology modeling, you can effectively build up the 3D structure of your protein. Okay, this would be the simplest and easiest way if everything is in your favor. Okay, well, say for example, now you end up in a situation where there is no structure of any homologue that has been experimentally determined, right? तो इसके दो जवाब आ सकते हैं, या तो उसका ये जो होमोलॉग अपन ने आइडेंटिफाई किए हैं यहाँ पर, if the structure of one of the homologue is already determined, और it is possible कि इनमें से किसी का भी स्ट्रक्चर अब तक डिटरमाइन नहीं हुआ होगा. None of the homologous structure might be there in your PDB, right? In such a case, it is not necessary that you end up your project. Okay, there are alternative ways that you can adopt, which I will just introduce you, and using this alternative way you can end up in you can end up in successfully predicting the protein model. Okay, so in such a case, where कौन से केस में, when you are sure that there is no homologue of any of the structure available in PTB. Okay, in that case you would first go and detect or predict the secondary structure of a given protein sequence. Okay, so well, say for example, now what we are going to do is that our objective is to figure out ki, okay, this is the sequence of what, homoserine kinase from Leishmania major. So now well, let us assume that no any structure of any homologue is available. So we would have to take this route now. Okay, so by this route, the first thing that you are supposed to do is to perform the secondary structure prediction. Okay, so what exactly do we do in secondary structure prediction is that we already know that there is going to be certain sequence over here. In our case it is P, G, A, R, D, E, S, K, F, G, such would be the sequence, right? So in secondary structure prediction, what our objective would be to figure out if this particular amino acid residue adopt what kind of secondary structure. So how many secondary structure do you know so far?
So
Yes,
two helix, alpha helix and beta.
ठीक है, there can be alpha helices, there can be beta sheet, okay, there can be turns, there can be loops. Okay, loops are also called as coiled coils or elongated, or you can say इसे अपन coiled coil कहेंगे. Okay, so these are possible for secondary structure that might be present in your protein. Okay, like what, helix, beta sheet, turns and loops. Okay, so the objective of performing secondary structure prediction would be to assign what each and every amino acid residue in your in your sequence might have what kind of secondary structure. Okay, well, say for example, if these first three residue or four residue might form helix, then is there any turn that might be adopted by these three residue? Then it is entirely possible that the next residue can be beta sheet. Okay, so assigning the secondary structure to individual amino acid residue would be the job of secondary structure prediction. You would try to predict what might be the secondary structure in your given sequence whose structure is needed to be predicted. Okay, is it okay? Okay, so by the
Yes.
end of the secondary structure prediction, you might end up with data. Like, we'll say, for example, in homoserine kinase of Leishmania, well, now I think that there are four helices. There are, we'll say, for example, three beta sheets. Okay, and there are, we'll say, for example, four loops, and there are 7 turns. Okay, you can get such information about what might be the secondary structures that are present in your sequence whose structure is needed to be determined. Okay, is it okay? यहाँ तक सबकी समझ में आ रहा है क्या?
Yes, sir.
Yes, sir.
Okay, so now once you have this information that my protein of interest might contain 4 helices, 3 beta sheet, 4 loops, 7 turns. Okay, then what you do is that you further proceed for fold recognition, right? So fold recognition का ultimate objective क्या रहेगा? To figure out if there are certain proteins which might contain the 4 helices, 3 beta sheet, 4 loops and 7 turns, right? So you can say that my protein can adopt that fold, right? Because you know that proteins are conserved more at the structural level rather than at sequence level. Okay, we have already demonstrated that in case of hemoglobin and myoglobin, उसके बीच में sequence similarity कितना था? बहुत कम था, but the structures were highly similar. So it is entirely possible that there might be certain fold which might have diverged quite earlier but might perform the same function, right? And this fold might contain similar secondary structures, right? And it is entirely possible then your sequence might adopt that particular fold. बरोबर है?
Yes.
Are you comfortable with this assumption?
Yes, sir.
Okay, so that is what we do in fold recognition. Okay, we try to figure out a particular fold for a given sequence. Okay, so this would be one of the topic that we are going to discuss. Okay, so now for this particular sequence you have determined that this is the fold, well say for example. Okay, this is certain fold of say certain protein that you have detected ki my sequence might fold up like this. Okay, then you have the same situation as homology modeling. Now you have a sequence and you have a particular fold, then you can use, well say for example, sequence structure alignment and perform homology modeling and end up generating the structure. बरोबर है? Is it okay?
Yes.
Sir.
Okay. Yes, sir. So now we'll say for example you do not identify any suitable fold for a given set of secondary structure that you have identified. Okay, then in that case you have, well say for example, failed over here. Okay, you were unable to predict the fold. In that case the answer is no. Then remain only one method by which you can determine the structure of a given protein. We are talking about this, and that can be done using something called as ab initio or something called as ab initio structure prediction. Okay, if nothing works, if you figured out that there is no homologue in PDB, you cannot perform homology modeling, then you attempted to perform secondary structure prediction and you attempted to identify a suitable fold, and yet you fail to identify that suitable fold, then only one option is remaining, is to predict the structure of the given protein ab initio. Okay, so in today's and subsequent lecture what we are going to do is that we are going to talk about the techniques like homology modeling, wherein you can directly predict the structure of a given protein if you have structure of homologue available. Okay, and you have got sequence available. Okay, you can use the technique like homology modeling. Okay, then secondary structure prediction, we are going to learn about secondary structure prediction. What are the methods, what are the types of secondary structure prediction. Okay, we are going to learn about what is fold recognition. Okay, what softwares are there for recognizing the fold as such. And then there would be a big portion in which we would be determining the structure of a given protein based on only sequence. We are not going to use any homologous information, or we are not going to use any information of its relative or its homologue in determining the structure of the given protein. Okay, we are going to generate the structure from scratch. Okay, that is called as ab initio protein structure prediction or de novo protein structure prediction. You won't use any information of any kind, and you would try to deduce the structure of a given protein solely based on the sequence of the given protein. That type of method is called as ab initio structure prediction method. Okay,
Yes.
is it clear? So this is
Yes.
what we are going to study for next few days.